Hello there. So I'm Megan Forsalati, and today I'm going to be talking about effective population size and inbreeding depression. So as a refresher from last class, we were talking about genetic drift and how it relates to effective population size. And so now we're going to be talking about inbreeding depression and how it relates to effective population size, and then also why it's important for conservation specifically. So one thing to keep in mind is that both drift and inbreeding depression have pretty similar impacts on the genome overall and on effective population size specifically, in the sense that both genetic drift and inbreeding depression lower your effective population size because they make it such that your time to most recent common ancestor or the fraction of the genome that's identical by descent is much higher or lower and higher. So it basically means that you have a common ancestor that was more recent. And it also means that the odds that you're identical by descent are higher. So I will be elaborating on that later on in the presentation, but I just thought that this was an important distinction to bring out from the beginning. And then another important distinction, if you guys are remembering from last class, is that genetic drift is something that is stochastic and it's a result of random sampling. So every population has genetic drift all the time as a result of the fact that sometimes which organisms just happen to pass down their genes and which genes just happen to be passed down because of recombination and linkage and independent assortment. Basically, all of these factors mean that there's an underlying randomness to a gene and whether it gets passed down into the next generation. So because of that, genetic drift is something that you'll see in every population, whereas inbreeding depression is not something that you'll see in every single population. You will see it in a lot of populations that exist today. A lot of that is because of human activity. So inbreeding depression can be highly influenced by lowering the effective population size, because that means that the opportunity for you to mate with another individual who is not identical by descent with you is much less likely. So on top of that, inbreeding depression, so we talked about how genetic drift is always stochastic, so it doesn't necessarily have a positive or negative effect. Inbreeding depression, that's not the case. As I'll be talking about when I define inbreeding depression will consistently lower your fitness because it's defined by the reduction of fitness. So as a result of that, when you have genetic drift, that's not always bad, but when you have inbreeding depression, that's probably bad for your population. So after that refresher, now let's get into the actual structure of this presentation. So I'm going to be redefining and reviewing effective population size. I just talked about it a lot previously, but in case you don't remember from last class, I'm just going to give a brief description of what it is. And then I'm going to be defining inbreeding depression. I'm going to be talking about how we measure it, why we use it. I'll also be providing a little bit more of context from the previous, based on what we learned in the previous class about how all of these concepts relate together. And then I'm going to be previewing why these concepts matter for conservation practices and give a brief summary of findings from case studies in our readings. So for our discussion, I have these points up. They're not necessarily requirements or anything. I just thought that they might be helpful in case we run out of things to talk about, maybe talking about these different things. However, I'm sure, and I saw that you guys had a lot of really interesting questions on the discussion post, so I'm not too concerned that we're going to run out of things to discuss. So defining effective population size, what actually is it? Well, if you take your current population and you simulated true random mating, the effective population size is essentially whatever size of a population under those conditions would experience the same loss of genetic variation from genetic drift, from random sampling of who gets to breed, et cetera, as your current population would. So the census population size is the actual size that scientists record, and effective population size is pretty consistently less than the census population size. This is because populations will vary in size over time, which means that the effects of drift will be different in different moments in time because there is variance in reproductive sex. So which organisms are going to mate and why they're going to mate is going to be different. And then also because of overlapping generations. So a lot of times you'll have age structure in your population So usually younger organisms, the youngest can't breed, younger ones can breed, and then older ones can't breed again. So you have all of these different roles that individuals are fulfilling. And when you measure the census population size, you're not necessarily measuring reproductive individuals. So because of that, a lot of times, how many individuals can actually breed in your population is going to be much less than how many you'll actually see. And we can measure all of this in the genome based on types of models that have been talked about in a couple of the papers, and also some of the fancy words that we learned about in class. So Bayesian or maximum likelihood or Markov models. So those are all kind of used to estimate effective population size. So inbreeding depression. Inbreeding depression is, as I was saying in the beginning, the overall reduction of fitness from consanguineous offspring. So we don't care about inbreeding if there's not inbreeding depression. You need a reduction of fitness for that to actually matter and mean anything. But the thing is that inbreeding depression is very common. And fitness is such a vague concept because it basically relates to your ability to survive and reproduce, that even though it may not seem that a population has lost fitness, it may have actually lost fitness, and we just don't know until there are downstream consequences down the line. So keep that in mind as well. And inbreeding depression is typically measured with traditional genetic methods, such as pedigrees. This is where a lot of our ideas about inbreeding came from. And then there are also these new high throughput sequencing methods, and these can either be mapped or unmapped. So inbreeding depression generally reduces hybrid vigor or heterosis, and it increases the proportion of harmful recessive alleles. So I'm going to be talking about the ways in which this happens. However, to kind of give some background with hybrid vigor and harmful recessive alleles, as we talked about in last class, there are phenomena such as genetic hitchhiking. So what can happen is that if a gene isn't explicitly causing harm in the population, then it can sometimes still gain substantial prominence, or it can stay maybe not at fixation, but at some sort of an unstable equilibrium within that population. So it's not going to get culled out directly by natural selection. If that allele is recessive and it's rare enough in the population, then it's unlikely that that allele is going to get removed by natural selection, just by virtue of the fact that the odds that you mate with another individual who has that allele are very small. So what that means is that there are a lot of recessive alleles going around in a population where if you were a homozygote for those traits, you would actually have a reduction in fitness. And what can happen is that when you're mating with individuals who are more closely related to you, you're more likely to inherit that rare recessive allele because you both definitely had an ancestor who had that recessive allele in order for it to be passed down to both of you. So if you and your sister or you and your brother both have that recessive allele and then you mate with each other, your offspring are probably going to be are going to way more likely be homozygous. So it would probably be under a Mendelian trait, a 0.25 chance that your kids would have it if you guys don't even have the trait. So that's actually why a lot of times when people are going to be having kids, they talk to geneticists to get pedigrees drawn, that way they actually know whether or not it's more likely that they're going to end up having offspring who inherit this rare allele, particularly if they come from certain genetic backgrounds, where we know that it's more common from inbreeding events in the past for people to have these harmful recessive alleles. And then hybrid vigor is basically the opposite in this sense, because it's getting reduced. The number of heterozygotes is going to get reduced, and I'll be talking about how that happens later on. But when you inbreed, you have less heterozygotes produced in a given generation. And that means that as a result, you have less opportunities for hybrid vigor to be displayed. An example of hybrid vigor would be sickle cell anemia, where when you inherit the sickle cell allele, but you're a heterozygote, you have a greater fitness advantage against malaria. So sometimes when you inherit a heterozygous trait, because of the amount of dosage that you're getting from that gene, from that allele, you are actually getting enough where it's healthy and it's helpful rather than harmful. So with sickle cell anemia, sickle cell disease is a disease where your cells form sickles if you have both copies of the sickle cell allele. And the issue is that the sickle cell red blood cells that you get as a result, they don't actually, they're actually pretty resistant to malaria. It's harder for the malaria parasite to infect you if you have red blood cells that have that shape. And if you have just one copy of that allele and you're otherwise healthy, then your cells are slightly deformed to the point where you can actually avoid getting malaria as easily, but you don't have all of the negative consequences that come from having sickle cells in the first place. So having sickle cells can be extremely harmful and it can cause a whole slew of health issues. So the thing is that one of the reasons why we think that this is such a common phenomenon, and there are actually multiple genes that can cause sickle cell, is because of the fact that malaria imposed such a strong selection pressure, and you have this hybrid vigor phenomenon. So you wouldn't be able to have heterozygotes who can survive malaria if you had a lot of inbreeding in your population. So that's kind of why inbreeding matters in that context. And maybe sickle cell anemia is a bad example because you do have harmful alleles too, but there are plenty of traits out there where having the heterozygote copy is an advantage. And maybe it's not necessarily, maybe it's neutral in the case of the recessive alleles. So here's inbreeding as a process. As you can see from the figure right here, you have these two siblings over here who end up mating with each other. And so because they mate with each other, they have these little black stripes, which are reflecting regions of the genome that were inherited from one common ancestor, or which are identical by descent. We talked about this in the context of population genetics and genetic drift last class. So the thing is that inbreeding, being identical by descent, that can be because you inherited something from a common ancestor, and in a population, that's going to be pretty common, but the time from which that occurred is going to be quite different. So for one gene, maybe being identical by descent isn't that surprising, but for this massive chunk of the chromosome, that's extremely unlikely, and it can probably be actively harmful, because that means that you are a homozygote for every single one of those traits. And that means you don't have any heterozygotes here. So as you can see in this plot here, if you were to imagine, say, this graph, you just have this giant chunk of the genome where there are no heterozygotes. And as I said before, with hybrid vigor and with the rare recessive alleles, that means that you both have less hybrid vigor and you have more homozygotes that probably have the recessive alleles. So this is kind of a mechanistic explanation for what I was talking about before. So we don't have it fully fleshed out why inbreeding depression occurs. Like, it's not as though we can just have a list of genes and be like, you will be inbreeding depressed here and here and here if you breed here. We don't have that consistent of a framework for understanding inbreeding depression, but we do have a rough idea that phenomena such as this are contributing to it. So there's definitely a lot more room for research at the mechanistic level for these types of things. So just keep that in mind for future research, that such things might be necessary. So returning to pedigrees, as I had mentioned 2 slides ago. So FP is an inbreeding coefficient, and it measures the probability that 2 individuals will be identical by descent based on this known pedigree. And a locus, as I said before, is identical by descent when you have these 2 homologous gene copies within it, which were inherited from this common ancestor. So the term identical by descent is a population genetics term. It comes from the Wright-Fisher model, and so with pedigrees, we're typically assuming that there was a time when none of the loci were identical by descent. Population geneticists use a statistic known as F rather than FP, and that also comes from Wright. And so this measures inbreeding in a population. So the problem with pedigrees is that there are a slew of impracticalities that come with using them. So you have linkage, and linkage means that individuals with identical pedigrees, because during meiosis there was this crossing over process, that the chromosomes that are being passed down to each individual are going to be different. So that's why you're different from your siblings, for example. So because of linkage, and because linkage is random, it means that when you're using this statistic, you're the data that you're collecting within the pedigree isn't a complete picture of what's actually going on, because it's not just like, oh, my parent has this gene and so therefore I will inherit it. There's also this aspect of it might get lost because maybe you don't get it from the right chromosome because that one crossed over at some point. So you might not get the allele. And so if we go back 2 slides to this figure right here, you can actually see that right here, where you have these two, this individual is super inbred. They have no heterozygous, they have like almost no heterozygosity in this entire chunk of the chromosome. And yet this individual here is equally inbred in the context of they have the same amount of ancestral inbreeding that has actually occurred. But in terms of how inbred they are in their genome, they're not that inbred. They only have like a little stripe that is showing that there was this inbreeding event. So it's a very stochastic process. And so that's an issue even when you're just measuring the whole genome. But when you're measuring it on a pedigree based on like phenotypic expression a lot of times, and maybe you have the genotype, but maybe you don't, that can be a huge issue, because you could get numbers that are just absolutely crazy and you might not be able to figure out what's actually happening with the pedigree. And then you also have this limited depth. So this kind of goes in with my point on how it's a biased estimator of F. So as you saw in the reading, what happens is FP is always going to be less than the population level F. And the reason for this is that when you assume that in a pedigree there was no inbreeding beforehand, that assumption is generally very wrong. It's usually the case that if a population is inbred now, at some point in its evolutionary history, it was also inbred. So a lot of times pedigrees, we only really started documenting pedigrees in like what, the 1800s, the 1900s. That's only 200 to 300 years of data. That's missing a giant fraction of the history of that population. A lot of populations nowadays, like they've been around for thousands, if not millions of years. Well, maybe not millions, because they definitely changed. But the idea still holds, is that the amount of information that you're getting relative to how old this population actually is such that you're really sampling only a tiny fraction of how much inbreeding there would have actually been in the population overall. And then finally, there's this point about FP being difficult to obtain. Pedigrees are really difficult to get, because to have a pedigree, you need to know how every individual in the family is related to one another. And unless you have a captive population with a small size, that can be really difficult to keep track of. Even with humans, we typically only get a pedigree that can be 5 generations at most. So it's very difficult to get this information. And not only that, you can't get a lot of information out of it. So that's why switching away from pedigrees was a huge advancement for conservation biology. And that's why I'm going to be talking about population level thinking and the F statistic. So FIS is just another measurement of F. It's also commonly used, which is why I included it here. And in general, F is measuring the relative amount of heterozygosity, how much it's deviating from what you would actually expect. So I have this formula here, which is a rearrangement of the formula back here, which is talking about F equals HE minus HO divided by HE. FIS is measuring a very similar thing. And so when we're looking at unmapped methods, what people will typically use is multi-locus heterozygosity, excuse me. And this is the proportion of genotype heterozygous loci. So as you see on these figures, this is just a plot of the distributions of different populations and how much of their distribution is heterozygous versus not. So as you can see in figure B, you have a much more disjoint distribution, and there's also a lot wider of a distribution, which suggests that and a lot of this wider distribution is located in lower regions. So, but basically just in general, this is showing that you could have different amounts of heterozygosity in different populations. And we use H, which is the true proportion of heterozygous loci across the genome. We basically get a statistical estimator for that in order to calculate F. So using this formula here, if you have an estimate for inbred versus non-inbred individuals and how inbred they would be, you can calculate F directly using this equation. And one thing that's important to note is that the expected heterozygosity of the population as a whole is negatively related to F. So as F increases, you're actually going to have a lower H because it's 1 minus F. So that means that basically the more inbred you are, the less heterozygous you are. So many studies have found that other estimators for F and MLH itself are robust for measuring F, and so scientists may employ these different methods if they want to weight certain types of evidence more than other types of evidence. So that's actually something that you saw in one of the papers we were reading on Tasmanian devils, is that the researchers there were using a statistic that basically just overweighted rare alleles to detect inbreeding depression. So that's just an example where maybe heterozygosity isn't the only information that you have available to you, and maybe you want to use other information as well. Well, as long as you're estimating heterozygosity overall, you can do that. So, and it'll all basically give you the same information. It just might be more appropriate for the kind of population that you're looking at. So I have a brief note in here about a couple of other high throughput sequencing methods that are unmapped. So these are just things that you might want to know if you want to study inbreeding depression. So you have microsatellite analysis-based estimators. That's what we saw with the Tasmanian devils. And then you also have FGRM, which are these diagonal elements of a genomic relatedness matrix. If you ever see a figure that looks like this is a genomic relatedness matrix, and the diagonals of it are what would be correlated with MLH. So if researchers are showing you a figure like this and they talk about the diagonals, then you'll know that they're measuring how heterozygous the population is. And then here's some high throughput sequencing map methods. So this was talked about pretty extensively in the readings. And the reason why map methods are kind of seen as the way of the future is because here you don't just have an idea of how heterozygous or homozygous the population is. You don't just have F. You also have an idea of what's going on structurally within the chromosome. So which regions of the chromosome are more likely to be inbred than others. And so that can give us a lot more information for any type of research. So that would be pretty important if we wanted to understand inbreeding more broadly. And it just gives you more information about the population. And using these models, you can also figure out NE a lot more easily, because you can actually see which regions of the genome and how those regions of the genome are more or less inbred. So blends of homozygosity are basically just the opposite of heterozygosity, where it's like these regions of the genome that have a lot of homozygosity. So they have very low heterozygosity as a result. And when scientists are considering these, they're also obviously accounting for sequencing error and mutation rate. I just included a side note on this because sometimes the polymerases are biased themselves. So certain mutations are actually more likely than others. So, and the polymerases are the same machinery as the ones that would be making the mutations in the first place. So because of that, you always need to account for those when you're doing any sort of assessment on the genome. And then, as I said previously, we can use these Markovian or Bayesian models in order to figure out where homologous chromosome segments coalesce in a recent ancestor. So coalescence theory, I'm definitely not like trained in teaching that, but the basic idea of coalescence theory is that you're coming up with an evolutionary history for your genes. So instead of treating it like usually you'd have an evolutionary history for a species, but here it's like every gene has its own little tree. And so you're tracing back where the common ancestor is inside of your genome. So basically using that kind of a model, using effective population size, you can figure out where that coalescence event occurred. So this works because every locus is assigned to the most recent common ancestor. So in practice, we kind of have seen based on these models that the regions of the genome that have a lot of heterozygosity are ones where they diverged a much longer time ago. And that makes sense because that means that they had a lot of time for mutations to accumulate. Whereas those that don't have that lack heterozygosity because they're probably more inbred. So that means that their most recent common ancestor was a lot more recent. We talked about identical by descent. So it's kind of very similar. It messes up the models here because it makes it so that the most recent common ancestor of two individuals in that population will have been much earlier. So I actually did not talk about all of this. So these plots here were covered in the figure in the paper, and just to review them because I thought that would be helpful. These are different populations of wolves. So in this population of wolves, this individual is not very inbred. And the way you can tell that is because there's very high heterozygosity here across the entire genome and very few regions where you have high homozygosity. Maybe here in chromosome 9, but otherwise you just have these long strips of heterozygosity. There's a lot of genetic diversity here. Whereas in this Ethiopian population, you have much lower overall heterozygosity, but if you notice, you really lack regions where there's a lot of no heterozygosity at all. So this suggests that this population had a low effective population size for a long time. So it doesn't have that much heterozygosity overall. The population is more self-similar than you would expect from this population, which is highly divergent. However, at the same time, there's not extensive inbreeding going on, because if there were inbreeding, you would have like no heterozygotes at all. So any inbreeding that has occurred in this population happened longer ago and was probably a consequence of the population dropping a long time ago. So there's less genetic diversity, but it's probably not a population where it's of huge concern. This, on the other hand, is really bad. You have just these entire little stripes on the chromosomes that just have no heterozygosity at all. This is a very inbred individual that's being sampled. And this is what a lot of scientists would be very worried about if they're measuring inbreeding depression. The odds that this population is not healthy are a lot higher than the odds that these two are not healthy. So long run-up homozygosity means more recent inbreeding event. Those giant stretches of dips in homozygosity dips in heterozygosity are where inbreeding more recently occurred. If it's shorter, then that means that any inbreeding that did occur was more ancestral. Higher heterozygosity means less inbreeding. Lower heterozygosity means more inbreeding. So this is basically just an inverse of what I just said. So relating effective population size to inbreeding depression. How do these concepts relate to each other? Well, I took this from a paper that is talking about effective population size and in a population genetics context. So essentially, when you have an effective population size, if there is more mating than you would expect by chance, FIS or F is basically just a measurement of how much more predisposed the population is to non-random mating with a with an individual who's closely related to you than by random chance alone. So this is an inverse relationship, so NE goes down. So you basically multiply it by this function in order to reflect the fact that basically what's happening is the genes are coalescing earlier. So the time to most recent ancestor is being reduced. So this is just like one of the mathematical ways that people will adjust how NE relates to FIS. So if you want to look at the math, this is the math. Now, does a low effective population size mean you have inbreeding depression? No. A lot of other things can lower your effective population size. And it's also important to introduce the concept of genetic load. So having low heterozygosity, like that Ethiopian wolf population, doesn't necessarily mean that you're unhealthy because you're not inbreeding extensively, right? So historically, small populations have the opportunity to reduce what's known as genetic load. So genetic load, I don't know, it was definitely a term that was used in the papers. So basically what genetic load is in 20th century thinking about evolution, about populations, scientists were really confused because why is it that populations aren't reaching an ideal? Why is it that they have all these traits that just seem to be so disadvantageous to them in the wild? And one concept which kind of came from this is the idea of genetic load, which is that a population is kind of a compromise of many different factors that are occurring, both historical and selection. And so selection is really what ultimately you get out of population is a compromise of all these many different forces that are working at once. So because of that, it means that you're going to have all of this extra DNA that's actively hurting you or is like not helping you, that is resource intensive for you to still have, but your population is stuck with it anyways because they can't get rid of it. If you have a smaller population, it's more easy for you to get rid of this genetic load because it's just easier for all of those individuals to die because like you have less individuals, period. So it's possible that over long time spans, and this doesn't just happen with small populations, it also happens with large ones. It's just that in small populations, it might be a little bit easier, but you're also more susceptible to drift, which is an issue. So, you know, it's kind of a trade-off. But just in general, sometimes you can have small populations which are very healthy. And mildly deleterious harmful alleles, those can still linger after demographic recovery. So even if you reduce your genetic load, so reducing your genetic load is basically the opposite of this. So it's possible you can reduce your genetic load, but it's also possible that you have all these extra alleles that are actually harming you. So in order to decide between which of these you're looking at in a population, you really need fitness data. So you need to know, is this population doing okay in the environment that it's in? So keep in mind, though, that as I just showed on the slide, inbreeding depression does lower effective population size. You have less genetic variability, you have reduction of hybrid vigor, you have increase of harmful recessive alleles, and just that reduction of variability even can have downstream effects that you don't even know about because they're for pressures that are going to happen in the future. So as I was saying before, you do need historical factors to understand effective population declines. So that's where a lot of times you'll bring these models in to figure out what happened in the past. This is something that was done in the Komodo dragon paper, monitoring how its population has had its effective population size change over time. And you can do this using SNPs and whole genome sequencing and correlate it with historical data such as climatic data like what we saw, and use that in order to get a more big picture story of what's going on. Now, this is with the caveat of this genetic adaptive variability concept. So this is what they're really worried about in Tasmanian devils, or even in Komodo dragons, is that we know that populations in the near future are going to be undergoing a lot of selection pressure because we are changing their environment so much. So they're going to have to respond to these. And inbreeding reduces their ability to respond to this by lowering their diversity. It's thought that when you have less genetic diversity in your population, that means that you have less mutations that might be beneficial in those adaptive situations, in those scenarios where selection pressure is high. So because of that, the population won't be as robust to change. So in summary, inbreeding's overall effect on the population and kind of how it relates to any more broadly, this is all a sum of how demographics have changed over time. Scientists consider this. This is really important for understanding how or why inbreeding might be more or less important. Remember that current effective population size can still be lowered by historical inbreeding. You have the strength of hybrid vigor in populations where heterozygous traits are more beneficial. That can be an issue. So if there's a population where having this one heterozygous allele is particularly influential in its fitness, maybe inbreeding depression will be a lot worse for that population. You also have the relative harm from recessive alleles. Some populations just happen to have more harmful recessive alleles than others, just by random chance, because a harmful recessive allele arises through random mutation. There's also the need for adaptation. So we know that populations will need to adapt in the future. So they need diversity. Because the thing is that selection can't create new alleles. It can't create new states. It can't create new traits. Selection doesn't create things, it only calls things. So mutation is really the thing that's driving the genetic diversity that would allow selection to act on a population. And when you get rid of all those mutations that have happened in the past, it means that you can't adapt. So that's what the last slide was mostly about. And then you have the overall genetic background, which I didn't really talk about too much. But the essential idea for this is that your population, your genome, is an interaction of many different instructions and a coordination of all these different instructions. So the overall genetic background, if you're more inbred in some parts, because of how those interact with other parts of the genome, that may be more or less influential. So that's kind of also something that you need to keep in mind, is that this isn't we think about things in a monogenic way because it's easier for our brains to think about things. But what's actually going on is that this giant coordinated act is being influenced by inbreeding depression. And so because of that, how big of an effect you're going to have, you have to think about it in big picture ways. You can't just think about it in terms of like one gene. So why these concepts matter for conservation? So I'm going to be giving some context behind the Tasmanian devil facial tumor disease in case some of you were confused by that. So the Tasmanian devil facial tumor disease is basically this disease where we think that the way it started pathologically is that Tasmanian devils have very low diversity in their major immunohistocompatibility complex. And this is the part of their immune system that recognizes self versus non-self. So we think that because of this low diversity, what happened was when one individual developed cancer and it bit another individual, that individual's immunohistocompatibility complex was not able to detect that that cancer cell came from a different individual than itself because the diversity was so low. So as a result of that, this disease is able to actually spread within the population because that overall genetic diversity is so low. And Tasmanian devils use facial biting as part of their mating. So this is really hitting the reproductive populations very hard. And Tasmanian devils before this were considered to be very healthy population overall. They weren't seen as something that was at risk of going extinct. And basically by 2008, the population of Tasmanian devils had declined over 60% from this disease, which started in the late 20th century. So this is not a lot of time for a population to be going through such dramatic declines. And as a consequence of the fact that these populations were in decline, there were trophic cascades. And foxes were actually able to be established in Tasmania, whereas historically they were competitively excluded because Tasmanian devils were an apex predator. And introducing foxes causes a huge slew of ecosystem effects. So that's why there's a trophic cascade as a result. And there's also evidence for evolution in the population and changing roadkill management. So the story is not all sad. Even though you have this facial tumor disease, there's also evidence that the populations have been rebounding and there are also vaccines available for them. So this is an example where inbreeding depression really matters because even though population declines occurred a long time ago and the population was seemingly healthy, that reduced adaptive variability that was available to the population meant that when this cancer cell was brought in, it made it particularly susceptible. So having less diversity can actually be an issue for your population. And so this study in particular was looking at inbreeding in a population that was not undergoing this disease, but was undergoing a fitness decline. There was a reproductive decline in the population. And so the scientists were trying to figure out why. And so what their models essentially found was that inbreeding depression that they measured in the population was correlated with the reproductive declines overall, but that many of the genes that were contributing to this decline were heterozygous alleles. So for some reason, the fitness decline, so their data was a little bit conflicting, but they still wound up going with the conclusion that inbreeding depression was an issue for the population. And so one thing that's important to note, if this seems confusing, is that even when you have evidence that there may be things other than inbreeding depression occurring, if you do have evidence that the population is undergoing a fitness decline and that this might be associated with some level of overrepresentation of homozygosity in the genome, then the solution to that is still going to be genetic rescue. It's still going to be introducing healthy individuals into the population. So this is why these scientists are still kind of publishing this data, even though it's giving slightly conflicting signals, is that they do find evidence that inbreeding depression is occurring, and that even if the heterozygous genes are the ones that by their models seem to be the most harmful, it could be that these are interacting with genes that are homozygous, and that's why they're causing those issues. And it could also be because maybe there's genetic drift, and so heterozygous traits are becoming more popular in the population. or they're more likely to be inherited, something like that. Maybe there's some sort of a rare allele that causes harm when it's in heterozygotes, and maybe it's now increased in the population, whereas when it's like recessive homozygous, it's not harmful. So you could have something like that going on too. So kind of the lessons that we can take away from this paper are that scientists monitor inbreeding a lot, and it's really important. And that kind of from Tasmanian devil facial tumor disease more broadly, it can cause huge issues in a population, even when it's not something that was originally, even when we don't think that the population is at risk. So in spite of this, there is a possibility for adaptation in the face of selective pressures. And so keep in mind that a lot of times you have all of these evolutionary changes that are occurring in like a whole system. And so microevolutionary processes can sometimes be beneficial or sometimes can be harmful. So as for the Komodo dragon population monitoring, remember that key takeaway I noted before, where there's that historical bottleneck that can have pronounced long-term effects, even though it's not necessarily apparent now. Looking at this figure now, you should be pretty terrified. I mean, these are Komodo dragons, and look at their genetic diversity compared to other reptiles, the tortoises, the crocodiles, the gharials, the alligators. Some of these are critically endangered, some of these are extinct. I mean, these guys are functionally extinct in terms of their genetic diversity if this plot has got things right. Sigma is just a measure of genetic diversity. We talked about it in last class. And so on this slide here, you can see that when the scientists actually removed runs of homozygosity from the genome, that this improved a lot. So basically, they're finding that there is heavy reduction in genetic diversity because of inbreeding in all their subpopulations of Komodo dragon camp-folds, and that the effective population size, they were actually able to reconstruct this over time. And the authors mentioned that there may have been a lot of gene flow that was important for population structure before human arrival and before climate change events, and that perhaps this restriction of gene flow is also leading to the populations being less robust. They were trying to make the argument that there's a Komodo National Park, but there are also parts of the Komodo dragon range that are not under protection, and perhaps they should be, so that we can maintain this reservoir and maybe even allow gene flow to be possible between populations that are separated from each other. So one thing to keep in mind with this kind of a comparison, which I did want to note, and I thought might be cool to discuss, is why they didn't compare Komodo dragons to other lizards. So one thing to note is that crocodiles, turtles, alligators, there are a lot of modern hypotheses about their evolutionary relationships are suggestive towards them being closer to each other to the exclusion of lizards and snakes. So birds, turtles, crocodiles, a lot of evidence supports them all being archosaurs together in a group that's separate from lepidosaurs, which is tuataras and squamates. So tuataras, lizards, snakes. So Komodo dragons aren't that closely related to crocodiles and alligators. So even though they're comparing these guys because of ecological reasons, because they're apex predators, they have small populations, they're endemic, they're endangered, they probably should have also, in my opinion, done a comparison with another close relative of Komodo dragons that maybe isn't necessarily as specialized, as unique ecologically, but is just closely related as a control. So that was kind of my thoughts on this paper, but otherwise it's really interesting that they have this reduction in genetic diversity, and also how much of this is effective population size because of human activity versus just virtue of being large-bodied. So that about wraps up everything I was going to cover. But there is good news, which I did want to include in my presentation. So the Bronx Zoo actually has baby Komodo dragons that have hatched. So you can actually see them in this video here. So it's not all bad, and I'm sure that having populations in captivity means that scientists will be able to better maintain genetic diversity overall in these populations. And you can actually see these guys at the Bronx Zoo. So yay. I'm not going to show the full video for this Zoom recording because it would probably be a waste of time. But yeah, they're babies and they like to fall from trees, which is not good because Komodo dragons should not fall from trees. For those of you who don't know, Komodo dragons eat their young. So if you're a baby Komodo dragon, the reason you're arboreal in the first place is because it's so dangerous to actually be on the island of Komodo, because your parents will just eat you. But like then these guys will just, like at the zoo, they'll just fall out of the tree. It's really funny in like their exhibits. So they're not that good at it. And so they're probably they would probably get eaten, the ones at the zoo. I've seen them. So that's basically all I have. So if you want to see my references on the next slide, you can. This is all the content, and then I have the images. So thank you for watching.